chapter 1 of the cardiac section. Sorry.
Right. I thought the clinical examination was a bit simple.
That's all we're covering today, Jeff. Just exam.
Yes, I thought. Then I looked under cardiac and I couldn't find a chapter 1 there. So.
Sorry, Jeff. You know, it's probably an area that you're quite familiar with.
I hope so.
Yeah. So we're covering the pathophysiology of heart failure, and in particular the neurohormonal mechanisms that contribute to heart failure. So the main things I want to sort of cover is, you know, we cover the kind of electrical physiology and where the electrolytes are going in the different cells of the heart and how that kind of stimulates contraction and coordinated heart function in normal animals last week. And this is where we just start to transition that into what's happening in abnormal animals. And next fortnight, we're going to do the treatment of congestive heart failure, or the treatment of heart failure. And we always say congestive heart failure because it's by far the most common form of heart failure. But heart failure can be forward failure as well as congestive. So the treatment of heart failure will encompass all of those. And we'll be talking about the little ion channels and stuff that we've been talking about these last few weeks. And obviously which medications will impact which of those normal physiology mechanisms. Let's start with what the heart's functions are. If we had to split it into 2 functions, what are the heart's main functions?
Helps with maintaining blood pressure and tissue perfusion.
Yep, excellent. Were they the 2? Sorry.
Oh, that was one I figured because kind of.
I'm only giving you one for that.
They're the same, really.
Sorry.
They are the same. Really.
Yeah, exactly. Yep. What about its other role in? It has to be able to receive the blood. So if we sort of think of the heart's role as being able to accept blood, and when we think of the atria as the sort of holding chambers, and they're the ones receiving the blood, and that's certainly part of it, but that blood has to be able to get into the ventricles fast enough and in enough volume to then be able to be pumped by those ventricles. So there's the diastolic function, which is the relaxation and accepting of blood. And then there's the systolic function, which is obviously the contraction and pumping of blood to meet perfusion requirements, which is then obviously so tightly controlled. So if we then look at those 2 mechanisms, how can we divide our definitions of heart failure?
Or forward and backward, I guess.
Exactly right. Yeah. And I think it's a really important concept to get that there can be a diastolic failure and a systolic failure. Now, most of the heart diseases and stuff that we see have a combination of both. So, for example, if we use mitral regurgitation, during systole, some of the blood's going forward out to the body, but a lot of it's going backwards as well. So we've got a degree of forward failure, we've got less cardiac output, but we've also got a degree of backward failure in that we end up with congestion and an increase in the holding systems or the venous system. And ultimately we end up with congestive heart failure, so backwards failure, predominantly, even though there is forward failure as a component of that. I was going to get my heart model, but I forgot it. Sorry. So I think it's really good to try and just like at this point talk about the different diseases that we see and what type of failure they cause in general, or think about, you know, will they cause a bit of forward, bit of backwards, mostly forwards. So if we talk about diseases of diastolic function, so diseases of receiving blood in the heart, can anyone give me any examples?
Like HCM.
Excellent. Yeah, good. How does that cause diastolic failure?
The muscle is too thick, the intraventricular diameter in diastole is small, so it can't receive blood.
Excellent, good. So we have decreased compliance of the heart muscle. It's stiff and hard, and it doesn't dilate and fill as well as it should. Any others? Lots of others.
Any condition that causes what you've just described, such as neoplasia of the heart muscle, which is rare.
Good.
And the other thing would be a pericardial effusion, which is not stiffening the heart muscle, it's just squashing the chamber.
Absolutely. That's a classic primary filling abnormality. So that primary function of the heart being able to receive blood, pericardial disease in general, so not just effusion, but what other ones?
Oh, traumatic pericarditis, I suppose.
Yep, traumatic or just infectious, reactive. There's mechanisms we don't understand. Any other pericardial conditions that might cause thickening or stiffening of the pericardium?
Pericarditis.
Yep. So inflammatory, and another big category.
I think Jeff said neoplasia already, I think, but
He did say neoplasia, yeah. So that's exactly, so I think Jeff was sort of, my interpretation of Jeff's comment was neoplasia that's compressing, so like a myxoma or something like that.
I was thinking more in terms of neoplasia of the actual heart muscle, making it stiff.
oh, okay.
That's why I said it was rare. So I didn't move the neoplasm into the pericardium, no.
But I think it's an important distinction.
Yes.
So what sort of neoplasias do we see in the pericardium?
Like, can you get lymphoma?
You do, good.
And
associated with asbestos.
Lymphoma?
No,
No.
neoplasia. Not in,
like,
I should clarify.
I can't remember the name, so it starts with M.
I can't think of it either.
It's to do more, is it to do more with the pleura in contact with the pericardium?
Yeah. Should I talk to you more, or should I tell you?
No, someone else.
Mesothelioma.
Good, right?
Yeah, that's the one.
Exactly, yeah. So if you see, it's particularly relevant on people doing echoes or T-fasts, the pericardium with mesothelioma or lymphoma it's spectacular. It's so thick. We see the same with chronic pericarditis, so it's not pathognomonic, but like so thick that we aspirate them and stuff, which you'd think is quite scary, but they're quite exciting diseases to see. So now going back to the heart muscle, neoplasias of the heart muscle, what do we see?
Hemangiosarcoma.
Good. Most common. Would that typically cause restrictive myocardial process?
No, but it could cause pericardial effusion, right?
Yeah, excellent. Any other infiltrative neoplasias of the myocardium?
Lymphoma again?
Good, excellent.
Well, lymphoma can be everywhere.
Rare, it's so everywhere. There's one of the differentials for hypertrophy on echo is an infiltrative disease like that.
Heartworm disease.
Will cause hypertrophy?
No, probably
Filling
not.
defect. It will definitely decrease diastolic function, ability for blood to flow into the ventricle.
I was thinking more of that. No, sorry, I'm off track.
No, it's good. That's a really good example of an infectious disease, which I didn't actually have on my list. So that's a really good point. It's absolutely a great example of a decreased filling failure. Something I saw on the weekend, which was so cool, a dog with bronchial carcinoma. And they asked me, I got called in on emergency weekend before last, and they asked me to do an echo on this dog, and it had a big tumor in its left ventricular myocardium, just a big like hypoechoic tumor. It's a really atypical location to get a hemangiosarc. They're normally right-sided, right AV junction, right auricle. And I looked it up, and there's reports of metastases, particularly from the lung, because the blood flows from the lung into the left side of the heart, and they tend to metastasize to the left ventricular free wall in the like apical third of the left ventricular free wall, which is exactly where I saw it in this dog. So I obviously didn't get histo or anything, but metastatic neoplasia as well should be on our list. So we've got pericardial disease, we've got neoplastic and inflammatory infectious processes, which will decrease filling. What about congenital abnormalities?
Congenital abnormalities causing any failure or
Causing
just
decreased ability for blood to get into the ventricles.
right? Well, right AV stenosis.
Excellent. Yep. Only right?
Oh, well, yeah, leftward as well. Yeah.
Yep, exactly. Yeah. So mitral or tricuspid stenosis will absolutely decrease the ability of blood to get into the ventricles and therefore obviously cause a forward failure, but it will be primarily a filling failure, a failure of that sort of aspect of heart disease. So what about diseases that decrease cardiac output or cause a forward failure? Let's start with congenital because that's where we finished.
Like your like a VSD.
Yeah, it will.
It's going to be going left to right.
Yes, very good. Excellent. So that will absolutely decrease cardiac output. Tell me how that's going to impact venous return.
I guess it's going to increase pressure in the right side of the heart. So you should get, it'll be more congestion with venous return in the right atria. It's a guess.
Yeah, absolutely. So we're going to get increased preload. So the blood that should be going out to circulation is going back to the right side, back into pulmonary circulation. So we get over-circulation of our pulmonary circulation and therefore increased preload in the left atrium. So we're actually going to get a volume overload on the left side. Excellent. So we've got left to right shunts. I'll put.
Sorry, is that volume overload on the left or the right?
It's actually volume overload on the left because the right will ultimately be overloaded too, but because of VSD, the blood goes straight from the left side of the heart, the aortic outflow tract, into the pulmonary outflow tract. So the right side of the heart doesn't actually pump more volume. So it's kind of the same volume. It's just pumping more blood into circulation, into the pulmonary circulation. So then you end up with increased venous return to the left and atriomegaly and obviously congestion in the left side of the heart.
Right, okay. Now, I wasn't aware actually that it actually went across from the left side into the pulmonary circulation. I thought it would have gone left to right ventricles.
It definitely does, but if you think about during systole, the flow of blood is going from the right ventricle straight out the right.
Yes.
So it's just sucking it straight into that pulmonary circulation.
Yeah.
And certainly the right ventricle will have some changes in that it will be having to pump hard to get more blood into there. But it's also going to be more at equilibrium with the left ventricle. So you'll see some changes in the right ventricle because of the increased pressure it's working under, but you won't necessarily get volume overload until it fails and decompensates.
Right.
What about other diseases where we get decreased outflow? So while we're thinking about aortic and pulmonic outflow tract.
DCM?
DCM, excellent. That's a really perfect example.
That's above the outflow tract.
But it's a perfect example of systolic dysfunction. So diastole is great. So it's filled right up, but it just can't pump blood out. So it's a myocardial failure. What about congenital valvular disease? Or subvalvular disease?
Like a stenotic.
Yeah, exactly. So the heart has to work so much harder to pump the same amount of blood through a narrower opening. So what are the diseases?
Like this aortic stenosis,
Aortic and
harmonic stenosis.
excellent. Good.
Would PDAs also cause like decreased outflow because it's more resistance, or am I just overthinking it?
Actually, less resistance on the left, but same mechanism as a left to right VSD.
Okay.
So you'll have increased blood flowing. So the left ventricle will pump really easily because most of the blood's going to systemic circulation, but some of it's going to the right side. So again, you'll end up with increased venous return to the left because the right side, the pulmonary circulation is over perfused.
Yeah.
But it should actually decrease resistance to outflow. But ultimately, it's a good point. So you're going to have decreased blood in the aorta because some of it's going to the left, but your baroreceptors should adjust for that. So your heart rate will go up and your stroke volume will go up.
And that makes sense as to why sometimes PDAs aren't addressed until they're older, right?
Exactly, yeah. So it's the size, the volume of blood shunting that determines when they're going to go into failure. So we've talked about the heart problems, most of the heart problems that cause decreased venous outflow. Decreased arterial outflow or aortic outflow. What about a systemic problem which would cause systolic failure or increased systolic pressure? I think I've worded that really badly. Do you understand what I mean?
You mean like renal disease?
Yes, but why?
Hypertension.
Hypertension, good, excellent. So similar mechanism to aortic stenosis, the heart's having to pump against increased, what's it called?
Vascular resistance, systemic vascular resistance.
Exactly. And when we're talking, another word for that that's used really commonly throughout the cardiac chapters, which you'll introduce is afterload. So the load that the heart is pumping against. So the load after the ventricle, essentially. So systemic hypertension, but also if we put the right side of the heart back in the equation, what other hypertension?
Oh, well, pulmonary hypertension.
Perfect. So increased afterload on the right side is pulmonary hypertension. Increased afterload on the left side is aortic systolic hypertension, systemic hypertension, An interesting concept that dawned on me, and I forget about it every few years, is cats with aortic thromboembolism. How is that going to impact the afterload in their heart?
You would think it would increase it.
Massively.
Yeah.
Knocking out like one whole way for the blood to go.
Exactly. So often these are cats with compromised hearts anyway. So 95% of the time they'll be cats with underlying heart disease. Often they've got left atriomegaly already, so they don't have very many compensatory abilities. And often they tip into congestive heart failure straight after they throw their clot. So there's going to be stress and adrenaline, but there's also going to be increased afterload, which then causes congestive heart failure, which tips them into congestive heart failure. So really important. Obviously, these cats come in with tachypnea because they're painful, but really important to be screening for congestive heart failure, obviously, in those cats. Going back to the DCM kind of systolic failure point, let's talk about myocardial diseases that decrease cardiac output. DCM's really common. The most common one, I think. What other
well, arrhythmias.
Good. That's what I'm getting at. Yeah. Thanks. What sort of arrhythmias will decrease cardiac output?
Atrial fibrillation.
Yeah.
Anything that's too fast so that there's no ability to refill.
Excellent. Yes, great.
So
Can you read the chapter, Alex?
No.
VPCs is, yeah.
Yeah.
But also sinus arrest.
Yeah, that'll stop. Yeah. So the point I want to make is that both tachyarrhythmias and bradyarrhythmias will decrease cardiac output. How does a tachyarrhythmia, and Alex alluded to this, how does a tachyarrhythmia decrease cardiac output? It should be increased, right?
You get poor contraction overall, I think. So, like Alex said, you just don't get a chance for the heart to fill before it can actually eject the blood.
Exactly. So is it poor contraction or is it poor relaxation?
Poor relaxation, yeah.
Good. Excellent.
Yeah, sorry, yeah, that's what I meant.
Absolutely. So if you look at a normal heart, as the heart rate increases, cardiac output increases almost linearly. So heart rate and cardiac output for you guys, go up linearly in a normal heart up to an absolute maximum where I think, anybody that's been in a gym and has seen your maximum heart rate is 220 minus your age. That's where your sort of the maximum is, where your ventricles just don't have time to fill between contractions. So each contraction just contains less blood and therefore you kind of plateau out when you get to the top. Interestingly, in diseased hearts or hearts that have remodeling or any myocardial abnormalities, it doesn't go up linearly. As heart rate goes up, stroke volume actually goes down because we get diastolic failure. We get inability to fill much earlier because there's often fibrosis in the myocardium in these patients. So particularly when heart failure is developing, what the body's first reaction to decrease cardiac output is to increase the heart rate, because the baroreceptors say we're not getting enough blood. We're going to increase the heart rate to compensate for that. So same as when you exercise, your body says, we need more blood to perfuse these muscles, and your heart rate goes up. That's just really automatic sympathetic nervous system reaction. And that will only work to a certain point in dogs with underlying heart disease, whereas in dogs with normal heart, their cardiac output just adjusts. Max, how did you go with the chapter? I don't mean to target you. Did you read it?
Yeah, I read it. Not recently, but I read it before. But
yeah. So there's a lot of detail in this chapter. It's really intense and it's only like 8 pages long, and it took me 3 hours to read. So I don't want to go into that much detail for the purpose of this tutorial, but I am, particularly for Max and Josh doing residencies, I am going to flag where there's something that I want them to read up on. So if you guys have a pen and paper, because a lot of the receptors and channels and stuff that we talk about in how heart failure develops are mechanisms genetically that have gone wrong to cause heart disease. And if you really understand why, it will really help your understanding of what tests, what treatment, what prevention are available. So let's talk about what makes the heart contract normally.
Like contraction rate,
No, force of contraction. Like, I just want to sort of talk about the elements of that quickly before we move on to the actual neurohormonal kind of adjustments that happen. Sorry, the sun in my eyes. So do you remember the pictures of the muscle? We didn't actually cover that last week, but the myosin, the actin and the myosin, and they overlap. Like this. Yeah.
Collated discs, whatever.
Yes, exactly. So say my thumbs are the discs.
Mm-hmm.
So they're the ends of the muscle cell, and they anchor my myosin and my actin together. So the myosin is bound to my Z disc, and that they then interact with each other under the influence of calcium to ratchet together and contract. So there's heaps of different elements of this that could go wrong to upset contraction and therefore result in myocardial or systolic failure, so failure of adequate contraction. So if we think of a couple of examples that are really relevant, particularly Josh and Max, do you know what myosin binding protein C is? Have you heard of that? No?
Never heard of it, sorry.
Never heard of that. So it's one of the proteins that binds the myosin filament to the actin filament and facilitates contraction. In cats with HCM, they have disordered or a genetic mutation in their myosin binding protein gene, and their ability for actin and myosin to move against each other is impaired. Now, when the heart can't contract adequately, it hypertrophies, it proliferates to try and make itself stronger. So we get disordered proliferation to try and strengthen the heart muscle, which results in concentric hypertrophy. So that's the bind between the myosin and the actin, which means it can't ratchet properly. So it's actually a disorder of contractility, which becomes a disorder of diastole, because the heart then gets so thick it can't relax. So that's sort of one point that I think is really important in muscle contraction. In humans, HCM is almost exclusively problems with the interaction between the myosin and actin, or problems at the anchoring points where the myosin and troponins bind to the Z discs. So they ratchet against each other and they actually twang off the Z discs. They're not well anchored, which results in hypertrophy of the heart muscle, because again, it's trying to stabilize itself. It's trying to generate enough force, but without being well anchored, it can't generate that force. So it replicates the heart muscles, causes HCM. So they're all sort of genes and proteins that are going to be really important as we get a better understanding of HCM in animals, and particularly in the ragdolls and Maine Coons, it's the myosin binding protein C that's the main protein we've identified as a genetic mutation. The other mutation we see quite often as a cause of genetic heart disease is in the way that calcium is processed in the heart muscle. So Max, sorry, can I get you to just tell me what the electrolyte movements are to stimulate contraction or action potential in a heart muscle cell? Do you remember?
Briefly from 2 weeks ago, I think the sodium get into the muscle cell and first thing, and increase the action potential to the threshold. And when the action potential increase to the threshold, it open the sodium potassium channel, and potassium get in, and I think at the same time also open the calcium channel, in particular in heart muscle. So calcium rush in, and that actually resulting a sustained contraction of the heart muscle.
Very good.
That's the special part of the heart muscle, and once the contraction finish, the sodium get out from the cell again from leaking. Or is it actively pumping out?
Active, yeah. Because it's up its concentration gradient, so it has to be active, because you know the ECF is higher in sodium than the ICF. That was excellent, really good. So sodium rushes in, potassium gets trapped in there actually. So potassium is already high in there, but it closes off the outflow of potassium, and the calcium channels all open, facilitating calcium to rush in. And the calcium is the thing that results in that sustained contraction. So sodium might cause this and calcium causes this, and actually facilitates functional heart, like ejection, essentially, good sustained contraction. Without that calcium, or with defects in calcium cycling in the cells, you can't get sustained contraction, and you will end up with DCM or failure of the myocardium. a systolic failure of the myocardium. So this is a really important point because medications that improve calcium cycling or calcium channel function in the cells will improve both diastole and systole. So pimobendan does that. So really important one to sort of understand how that works. So where the calcium actually comes from, if you're just looking at a single cell, 10% of the calcium comes from the extracellular fluid, but actually 90% of it comes from the sarcoplasmic reticulum, little storage facility within the cell that just stores all the calcium. So you get that opening of calcium channels, little bits of calcium flow into the cells, and that then triggers the opening of these sarcoplasmic reticulum calcium channels, and all the calcium in the sarcoplasmic reticulum flows into the intracellular fluid, and that creates that sustained contraction. 90% of it comes from the sarcoplasmic reticulum from inside the cell. So then once the contraction's finished and the cell's trying to repolarize, it's relaxed again, the calcium needs to be moved back into the sarcoplasmic reticulum to create that potential difference across the membrane. So there's a few different elements along this point that are targets for therapy or places where diseases arise. So calcium flows in from extracellular fluid and then attaches to the ryanodine receptor on the sarcoplasmic reticulum. And that's what triggers the opening of the calcium channels in the sarcoplasmic reticulum and the majority of calcium to flow out. Then there's an active, what's he called? Calcium ATPase. So, there's a sodium potassium ATPase, an active pump that pumps the sodium and potassium back to their original concentrations in the membrane. In the sarcoplasmic reticulum membrane, there's a calcium ATPase pump that actively pumps all the calcium from the ECF back into the sarcoplasmic reticulum. And that's just got to happen between every single contraction of the heart. So relaxation of the heart depends on all the calcium being back in the sarcoplasmic reticulum, because otherwise you're still going to get a little bit of contraction. Contraction of the heart relies on all the calcium flowing back out of the sarcoplasmic reticulum into the ECF, and then back again, and then back again. So such an active process for sodium, potassium, but particularly for calcium, which is the one that's so important because we use it as a therapeutic target. That ryanodine receptor, which is the trigger for opening of the calcium channels. What's the disease? I'm pretty sure in humans, abnormalities of that ryanodine receptor are associated with DCM development. I don't know if it's been documented in dogs or not. I hope I wrote that down somewhere. Anyway, very interesting point for potential abnormalities of systole. Anyway, so that's enough for sort of basic physiology. I won't lecture you anymore. But they're the important points of like the actual sort of contraction. So what happens when we get initial forward failure of the heart? We've already sort of talked about it a little bit. We get decreased cardiac output all of a sudden, and then how does the body respond to that?
Increases heart rate.
Yeah.
Yeah.
Excellent. Well, it does. Yeah. That, but also I think it tries to increase systemic vascular resistance as well.
Yep. Excellent. How?
So the baroreceptors detect the change of cardiac output, and then they tell the brain, I don't remember where, but then that increases sympathetic tone. Great. Generally, which I think there's a great table in the physiology textbook that I don't have in front of me. I didn't bring it home, but it talks about the different like alpha and beta receptors that work on the blood vessels and on the heart as well.
That's exactly right. So it works on the blood vessels in the heart, but what hormone I mean, sympathetic nervous system. What hormones are we talking about?
Like noradrenaline,
And?
adrenaline.
Yeah, excellent. Good. Where's adrenaline produced? Sorry?
Adrenal gland.
Good, exactly, adrenal medulla. Where's noradrenaline produced?
It comes from the same place, but it's a conversion, I think, of, oh no, wait. Same place.
Same place, most of it. What about when it's used locally as a neurotransmitter?
Pass. I don't know.
The sympathetic nervous system has two kind of components. One is circulating catecholamines, which trigger beta receptor or alpha receptor activation systemically throughout the whole body. But the other component is actually just releasing noradrenaline from the nerve endings locally. And that's why you can get changes, like you can get an increase in heart rate in the presence of vasodilation potentially, or like urethral dilation at the same time as an increase in heart rate kind of thing. So it allows a little bit more localized sympathetic activation. So noradrenaline is the precursor for adrenaline, and the only cells in the body that have the enzyme, I think it's called, I don't know what it is, the thing that converts noradrenaline to adrenaline is in the cells of the adrenal medulla. So adrenaline only comes from the adrenal medulla, but locally throughout the body, the nerves of the sympathetic nervous system using noradrenaline as a neurotransmitter can titrate or fine-tune the sympathetic response. So it can be a little bit more localized than the whole systemic flood of catecholamines.
Adrenaline is an alpha and beta agonist. I've just forgotten what noradrenaline is.
It's both actually as well. It's non-specific. Yeah. So in the context of heart failure, is this a positive thing or a negative thing, this adrenaline, increased catecholamine, increased sympathetic nervous system?
It's negative.
Negative long term, isn't it?
Yeah.
Why?
Because it's going to increase systemic, we're trying to increase systemic blood pressure, so increase afterload.
Good.
So the heart's trying to kind of combat that. And then overall, it's going to add to increased preload as well and congestion.
Yep, excellent.
It also, like as a response, tries to kind of keep blood pressure centrally, so you get less blood flow to organs like your kidneys and gut, so you can get ischemic injury.
Great.
That's
excellent.
right.
Yeah, no, that's really good. Excellent. Over time, does the body have a way of decreasing the impact of increases in catecholamines?
I don't know. I don't think so, but I don't know.
Well, it's interesting. The body gets desensitized to catecholamines. So over time with chronic exposure, there's a down regulation of adrenal receptors in the myocardium. So the beta 1 receptors are decrease in number. And they're also a bit tricky. So every receptor on the surface, so noradrenaline hits the receptor on the surface, the receptors are linked to the intracellular signaling molecules. So with chronic exposure, the cell just like takes the intracellular signaling molecule away from the receptor. So the receptor just doesn't do anything. So adrenaline or noradrenaline bind to the receptor and it just doesn't do anything intracellularly. So it's called uncoupling with the receptor. So you ultimately, if you give a dog with congestive heart failure adrenaline, they've got less capacity to respond to it because they've got a decrease in the number of receptors, but also a decrease in the responsiveness to binding to those receptors. So ultimately they become less dependent or responsive to catecholamines. In humans, noradrenaline levels in circulation correlates really closely with severity of congestive heart failure. In dogs and cats, it's a little bit harder. There's so much more variability in anxiety levels and endogenous catecholamine levels in a hospital setting. But there is correlations with heart failure and increases in adrenaline and noradrenaline. Okay, so after adrenaline and noradrenaline, so after the sympathetic nervous system gets activated by your decrease in cardiac output, what is impacted next by your decrease in cardiac output? What system?
Like your kidneys?
Yes, excellent.
Yes.
What's going to happen?
It's going to activate RAS.
Yes, excellent.
I was waiting for us.
Let's talk about RAS. Okay, so can you tell me exactly the process of decreased renal perfusion and then?
Me or Alex?
Anyone.
Go, Josh.
Someone else? I feel like I'm talking too much.
No, please don't.
Never.
I think all the time, Jeff. Decreased perfusion of the kidneys will activate renin release.
Mm-hmm.
And I'm sorry, I can't remember the exact, like every part, so someone else might be better at this, but basically like angiotensin gets converted to angiotensin 2, which then does some things and stimulates aldosterone release. So that part, I need to go through that again.
It's good.
It's
good.
But at the end thing is aldosterone is released, which in higher quantities, and then that increases sodium retention, which increases fluid retention, which increases systemic blood pressure, which then again affects afterload and preload. Excellent.
So exactly right, and those are, you've mentioned all of the hormones. Sorry about the printer, I don't know what's going on. You mentioned all of the relevant hormones there. I think the important part is to, just because of therapeutic targets, to highlight that angiotensinogen is converted to AT1 by your renin, and then angiotensin 1 converted to angiotensin 2 by what?
Angiotensin converting enzyme.
Excellent, very good. And why is that significant?
Because you can use ACE inhibitors to stop that conversion.
Very good, excellent. So renin does stage 1 conversion, ACE does stage 2 conversion. Excellent. So angiotensin 2 you mentioned has what effect on blood vessels, Max?
Increased vascular tone.
Excellent. And
increase the efferent artery into the kidney and dilate the efferent, which is the exit of the kidney.
Excellent. Why does it do that?
Trying to increase the renal pressure glomerular. Well, I say it's renal perfusion. Yeah.
Yes, good, exactly, excellent. And you mentioned it triggers aldosterone release. Where from?
From
the adrenal cortex.
Excellent, good. What else triggers aldosterone release?
Apart from that RAS, tumours.
Yes, good, excellent. What other physio? So if I did a blood test, what electrolyte might?
Oh, potassium has a quite a big influence on that. Yeah.
Exactly. So if I'm hypokalemic, my body wants to produce aldosterone to conserve, to reabsorb potassium from the tubulars. And there's one other stimulus of aldosterone secretion, which is not so relevant to congestive heart failure, but it's ACTH. So when we're doing stim tests and things, we're not just stimulating cortisol, maximal cortisol secretion, we're also stimulating maximum aldosterone secretion.
Do we measure that?
Hey?
Do we measure that?
Sometimes we do renin aldosterone ratios to work out the degree of activation of that system, particularly in patients with like hypokalemia and stuff. Okay, excellent. So we know about the effects of RAS on heart failure. Retaining fluid has what impact on the heart function itself?
It has to work harder.
Yeah, exactly. Why? What's the word for there's an increase in
Volume, load. I don't know. Preload.
Preload. Good. So if you think about the effects of this system, it's going to cause vasoconstriction of the arterial system and actually decrease arterial volume, the volume in the arterial system and arterioles and things. But ultimately, it's going to cause an increase in volume in the venous system. So you get dilation of your venous system and overload of your venous system and decrease in volume of your arterial system. And that leads to what?
Well, if they're in congestive heart failure, it makes it worse.
Absolutely. Well, it tips them into it is the development of congestive heart failure.
Okay.
So that increase in volume retention would manifest as what measurement on our echo?
Intraventricular diameter.
Very good. Excellent. And?
The atrial size, left atrial size.
Good. Excellent. We just can't measure the right atrium as accurately. It should be a good measure, but we just don't get as good a view. And it's because it's lower pressure, it's kind of pushed out of the way by the left atrium. So we use left atrium as our marker of volume, which only works if the blood is flowing from the right side of the heart to the left side of the heart. Like you don't have pulmonary stenosis, you don't have pulmonary hypertension, you don't have any shunting through the from right to left or the through the lungs. Okay, so we've talked about sympathetic nervous system, we've talked about RAS. What about how this all impacts the hormones that impact kidney function? What are those hormones other than RAS ones? I think someone said something, but it's super quiet.
Antidiuretic hormone, ADH.
Excellent, very good. What's the other name for ADH? Say again, sorry. Good, excellent. Yeah. I asked that question because it was embarrassingly far through my studies that I made that connection. So just making sure everybody knows. So what does ADH do? And what makes it, what causes ADH production?
It comes from the, it's the pituitary, I think.
It is, yeah.
And acts on the receptors within the kidneys.
Mm-hmm.
And ADH increases fluid retention in the kidneys.
Mm-hmm. Excellent. So given that we know what it does, it causes fluid retention, what might trigger its release from the posterior pituitary?
Hypotension.
Yeah, good. Well, hypovolemia.
Yeah. Oh, yeah.
Yep. And
is it sympathetic nervous system?
It's actually not. So say we've got hypernatremia, what impact is that going to have on the osmolality of our blood? So
just to do with osmolality.
Yeah.
So it's going to increase osmolality.
Exactly. So increased osmolality or decreased volume will trigger ADH release. How does the body detect a decreased volume? Hypovolemia.
The baroreceptors again? Or is it? No.
That's deep because it's not, you can have pretty good blood pressure, arterial blood pressure, even in the face of like an acute bleed and a decrease in volume. It's actually not a pressure receptor. That's not true. It is a pressure
A
receptor.
stretch, is the atrial stretch receptor?
Yes, excellent, excellent. Yeah, good. So atrial stretch receptors fire continuously and that inhibits ADH. And then when we get hypovolemia and the atria shrinks down, there's less stimulation from those stretch receptors, which is what actually uninhibits ADH release. It's kind of a bit of a double negative concept. So when there's less stretch in the atrium, we get ADH release to increase volume. Does that make sense?
Yeah.
Excellent. So what does ADH do other than in the kidneys? Do you know if it has any effects in the blood vessels and heart?
Vasoconstriction.
Vasoconstriction, exactly. And it's actually a positive inotrope. So given in heart failure, should we have an increase in ADH or a decrease in ADH?
Decrease? No.
It should be, but it isn't. Because the atria are stretched. So we should be inhibition of ADH release. But it's actually the opposite. And I don't really know why that is. So just to be really annoying, the point here is that there's something about triggering ADH release that we don't know about. They think it might be low blood pressure actually causing baroreceptor, so decreased cardiac output, triggering baroreceptor feedback to the ADH release, but they haven't actually identified that pathway. So there's specific ADH receptor blockers that they're looking at as a therapeutic target, but they haven't found any therapeutic benefit for congestive heart failure at this point. What other hormones will impact renal function?
Was the ADH trigger anything to do with the natriuretic hormone, like the BNP and ANP?
Was the what trigger, sorry?
Was there anything that do with, like, I just can't think of a clear connection as in the natriuretic hormones that increase sodium loss. Does that actually trigger ADH production?
It doesn't actually trigger ADH production, but ANP and BNP, that natriuretic peptides, definitely impact renal function.
Oh.
So how? Do we know? The clue is in the name.
Increase sodium excretion.
Good, excellent. Yeah, so it's natriuretic, so sodium excretion. And then obviously the fluid follows with that. What impact would that have on RAS?
Probably activated, would it?
Beg your pardon, sorry?
Would it activate it?
It actually would make sense, but it actually inhibits renin and aldosterone. So it's in this context where there's decreased renal perfusion because of decreased cardiac output, it kind of counteracts the RAS system and does inhibit the renin and aldosterone levels in these patients. So it offsets that volume retention aspect, the RAS aspect of congestive heart failure. Are these levels, what's the clinical significance of, for example, BNP?
The respiratory sign, is it because of congestive heart failure because of this stretch of the atria?
Exactly, yeah. So it helps us. It's a clinical test that we can do to test the pro-BNP, which is just a more stable form of the B natriuretic peptide, which is produced in the atria when the atria is stretched, and also in cats with ventricular failure, it is produced in the ventricle as well. So the levels go up clinically when we go into congestive heart failure. So differentiating, particularly in cats, differentiating between respiratory and cardiac causes of tachypnea or cough is really helpful. I am actually going to have to wrap up. We didn't get very far through there, but I'm sure we're going to cover the rest of it in context with talking about the medications or management of heart failure. So that'll be the chapter. So it's the second chapter in the cardiac section, Jeff. We're going to do for the next fortnight.
Thank you, Anna.
Good luck with
thank
your
you so
cat,
much.
Anna.
Thank you.
Yeah, good luck. Thank you so much, Steph. That's awesome.
It's a good sign nobody's coming.